Excuse me, where is my slide? No idea.
Yep. So you are right here. So hold on. So yours should be right. This one. Okay.
This one.
Hold on. Let me open it as a PDF file. Come on.
Yeah.
Yep, then it doesn't like. Are you okay presenting on this? Because you have to like keep scrolling instead of using.
Oh.
Yeah, so like this is how you're going to have to scroll. Oops.
I prefer the PDF.
It's this one?
Yeah. So it doesn't like give you the option to open with.
It doesn't select this one. Yeah. Should select.
I'm just gonna open in here, so we'll do.
There we go.
Good afternoon, everyone. My name is Fangda. The first author, Zhenwei, cannot attend the conference due to the visa issue, so I present his work, Bench Effects Correction for Microbiome Data with Dirichlet-multinomial Regression. Microbiota play an important role in imitating our health. We can find them everywhere. For example, our mouth, skin, gut, or even blood. Their mouth is very huge. Taking our gut as an example, there are over 10 trillion gut microbiomes, and with over 1,000 prevalent bacterial species. They play very many functions, and they can provide nutrition, contribute to metabolic and immunological functions. Recent present matter genomics analysis function equip us with the probability to quantify microbiome data. With the decreasing cost of the sequencing, more and more samples are sequenced, especially with the emergence of the consortium projects such as the American Gut Project and the MAT-HIT project. We want to ask the question, can we pull samples from different cohorts and analyze them together? Integrating samples from different studies can improve statistical power and reduce cohort-specific bias. So can we pull samples from different studies directly? Here, let's look at an example. We collect for colorectal cancer metagenomic study, and these 4 studies are sequenced in the same sequencing platform. To virtualize the overall difference of the microbiome composition from different cohorts, we plot the first 2 principal coordinates in this figure. Each dot in this figure represents the microbiome composition of a sample. And we can see there is significant difference between different cohorts. I'm saying the blue one is this part, and I think the orange one is concentrated at the left-hand side. And we also use Priminova test to detect the difference, and the p-value is significant. Here, I want we call this the difference between different cohorts as a bench effect. The bench effects can caused by different platform, labs, reagents, or even different experiments. And here is an example of Bacteroid vagatus. It's one of the most abundant bacteria in our gut. We first, here is the density curve of the read counts. We first compare the two red curves. The difference between them is the difference between the samples. Here is between the controls and cases in the Australian cohorts. And then we want to compare the difference between the red curve and the green curve. This is the difference between two cohorts. We can find the difference between two cohorts is even larger than the difference between cases and controls. So the bench effect is very severe. It can even distort the association between microbiome taxon and phenotype, thus making it difficult to integrating studies directly. So the first target is to correct bench effect for microbiome data. And then we will identify association between microbiome taxon and phenotype after correcting. Actually, people have been long aware of the bench effects in high throughput sequencing data. But there are lots of methods focused on the gene expression data instead of microbiome data. And the microbiome data has its own characteristics. Here I summarize some characteristics between the microbiome data and the gene expression data. Let's focus on the second characteristics. Usually when we analyzing gene expression data, we assume the expression level of different genes are independent. On the other hand, for microbiome data, the read counts of the microbiome represents the microbiome composition. In other words, the sum of all taxa read counts is equal to a constant. Actually, the read counts of all taxa lies in a high dimension simplex. So we need to model the dependence between the read counts. Here I want to talk more about the effect of dependence. Here is a toy example. We have 6 species. The first row is a control sample. The read counts of each taxa is equal to 12, and the total number of read counts is 72. When the person suffers from disease and become a case sample, the first 3 species increase, and the total number of read counts is 108. When people want to compare the performance of microbiome composition, we will do the downsampling to make the total number of the read counts in the case sample is same as that in the control sample. But as a result, we can say the first 3 species doesn't change in the microbiome composition, but actually their amount is changed. On the other hand, the species 4, 5, and 6 decrease in the microbiome composition, but in fact their amount doesn't change. So we call the first 2 as false negative, and the last 3 as false discovery. So if we ignore the dependence between taxa, we will suffer from the increased number of false negative and false discovery. So let me introduce our model. We model the microbiome data, composition data, Yij, I for the bench number, J for the sample number, and G for the taxon number. Y satisfies Dirichlet-multinomial distribution with parameter gamma ij, and gamma ij has this form. We want to model the baseline for each taxon, and Zij is the phenotype we want to we conserve, and Xij is a p-dimensional confounding covariance such as age, gender, or even BMI. And delta ij is bench effect variable. We want to correct this effect. The Vij encodes the sorry. The Vij encodes the phenotype and the effect of the phenotype. And we want to detect the association between the microbiome composition and the phenotype. So we want to verify whether or not Vj is equal to 0. We conduct a Bayes approach. We pose a spec and slide prior on Vg. Kc is an indicator to indicate whether Vj is equal to 0. In the first condition, the tau is large, so it seems like Vj is not equal to 0, has a large variance. And in the second circumstance, Kcj is equal to 1, Kcj is equal to 0. Eta has a small value, so we assume that Vj is close to 0. And then we use Monte Carlo Markov Monte Carlo method to estimate the parameters. We want to threshold the value of Kc. If Kcj has a large probability be equal to 1, we say the taxon J is associated with the phenotype. On the other hand, if Kcj is equal to 0, we say this taxon is not associated with the phenotype. We use the median prob model and constrain the Bayes false discovery rate to detect whether or not the Kcj is equal to 1. We conduct some to analyze our performance. We conduct some simulation analysis. First, we compare meta-analysis with single cohort analysis. Our variable selection performance is evaluated by the precision recall and F1 score. We can say the darkest one, our proposed method is outperformance than the single cohort method. And then we do some sensitivity analysis and benchmark our method with some present meta-analysis methods. And we can say the darkest one is our method. First, we simulate data with different over-dispersion degrees. Our method is not sensitive to the over-dispersion and outperforms other alternative methods. And then as the microbiome taxon is more associated with phenotype, we want to check whether or not our method is keep a robust variable selection on this abundance of correlation between taxon and phenotype. So our method have a robust variable selection compare other method. We can say if there are more taxon related to the phenotype, we can say alternative method will suffer from more false discovery rate. And our method have a robust model variable selection. Here we apply our method to for colorectal cancer studies mentioned before. First, we want to test the goodness fit of our model. The correlation coefficient between estimate read counts and the observed read counts is 0.8. That's to say our method fits the real data well. And our proposed model also identifies 6 species significantly associated the colorectal cancer, and we found our selected species are also supported by some literature, including some biological literature and some sequencing researchers. To conclude, we propose a method to correct bench effect for microbiome data. And our method captures some unique characteristic of microbiome data, including the dependence and the over-dispersion of the read counts. In the simulation study and the real data analysis, our method fits the data well and reduce false discoveries. We also prepare for our package called BDMMA, and it's available on the GitHub. We want our method is be useful to analyze the large consortium projects, including the American GitHub and the MetaHit projects. So actually, this is my right position with the acknowledgement. The author cannot attend the conference. So thanks for your attention. Do you have any questions? Oh, this is his Gmail. If you have a question, if you more, please.
You were talking about the gene expression at the beginning when you introduced your work.
Yeah.
You said that gene expression has almost no zeros. So were you referring to microarray data or to sequencing data?
You mean I can extend method to some data with a lot of zero counts? You mean?
No, I just want to know. You had a table at the beginning where you were comparing microbiome data versus
Yeah.
gene expression.
Yeah.
And in that table, you wrote that gene expression has almost no zeros in there.
Yeah.
So I'm asking, what's the source for that? Is it microarray data, is it sequencing data, or what do you use for that?
Use microbiome read counts is something the microbiome composition data. Instead of gene expression data. Sorry. Is that answer your question? Please say again. I can catch it.
I just want to know, you said, I want to know on what you claim why there's almost no zero entries. Partly back to that slide where you summarize all of it.
You mean the real data have less?
The table where you had on the left side gene expression, on the right side the microbiome.
Yeah.
And there you say it's only a few zeros in there for gene expression.
Yeah.
And I just want to know, the gene expression, is it measured based on a sequencing or next generation sequencing methodology, or is it based on microarray data? That's the question.
Actually, I'm not familiar with the real data, how it's like. If you have this question, you can ask the author, actually. I'm not focused on this part of data, how the data is look like. Yeah. I'm sorry. Do you have any questions? Thanks.
Well, thank you.
Here, the last one is his Gmail. You can ask him. Sorry.